Good morning, good afternoon, good evening. I'm going to speak about harnessing the promising fruits of genomics-assisted breeding in, clonally propagated crops, specifically in yam. And why I say this, because yam is among many of the clonally propagated crops is one of the orphan crops where a lot of development has not happened. But over the last 5-10 years, there has been some great strides that has been made. So I'm just going to share some of the improvements that we have made in genomics assisted breeding in this crop. And this is how yam looks. This is not sweet potato that is being confused with, but this is how it looks. And this is how the crop looks when it is growing in its highest vegetative form. Okay, so I just want to say, I'm sure all of you know about what is clonally propagated crops. Mainly it is asexual, then they are highly heterozygous, long breeding cycle. Some of them have multiple ploidy levels, poor to no flowering or seed set. Self-incompatibility is one of the issues. Genetics have not been understood very well, and there are several pests, diseases, and storage problems, and of course, post-harvest losses. Coming to yam, what is yam itself? It's a Dioscorea species and is used as food and cash crop in West Africa mainly, then in Asia, Pacific, and the Latin America. There are about 300 million people in Africa that depends on yams. 6.7 million hectare land is being used for its production, and it gives about 58 to 60 million tons. It belongs to Dioscoreaceae family with 600 species, out of which only 10 species are used as cultivated ones, and a lot of species have not even been understood very well. It has got very high nutritional and medicinal values. If you compare most of the starchy crops, it seems to have one of the lowest glycemic index. Cash income of about $13.7 billion and exceeds all other staple crops in Africa, including maize, cassava, etc. It has got a lot of socio-cultural importance. It has got yam harvest, if you can see. The cultural is, if somebody wants to marry a girl, they have to give the largest yams that is available in the country to the family of the girl to marry that girl. Anyways, so and again, yam improvement, as I said, it's clonally propagated, is majorly dioecious, but it seems like the flowering is in kind of domestication or it is under evolution. So you can see occasional monoecious flowering as well. It has got very long breeding cycle, about 8 to 10 years, multiple ploidy levels. And one of the major constraint is we don't even know the ploidy levels or what kind of ploidy is there. Recently, with the whole genome sequencing of D. alata, one of the species, we found that it is a allotetraploid. It has got multiple species, as I said, poor to no flowering. Tuber dormancy is another issue. Several pests, diseases, and storage problems, and of course, post-harvest losses. So this makes yam breeding really, really difficult to generate new varieties. We all know about this breeder's equation to accelerate genetic gain, and this is also what we are applying in yam breeding to carefully plan the change the genetics of materials for a defined target environment, and then use state-of-the-art techniques to reduce error and speed selection turn, and of course, timely delivery of important varieties. Factors that influence the varietal adoption mainly, we did some studies in Nigeria as well as in Ghana, and we found that using participatory and gender sensitive tools to understand the traits and the criteria that farmer considered. So we are actually following the modules of excellence in breeding to actually define the pipeline for product development. And farmer or consumer surveys are being done, and we try to understand what traits define variety, etc. And we try to do trait benchmarking. So in one of the things that is very important in yam improvement is pedigree, because it's a clonally propagated and there is a lot of mislabeling and there's a lot of mixture, and there is also a kind of evolution or domestication going on between two species, which is Dioscorea rotundata and Dioscorea caiensis, along with the wild species like Prahensilis and Abyssinica. So there's a lot of mixture. So we need to understand the pedigree relationship, and also we need to understand which particular breeding line or land race is contributing to those that pedigree. So this is something we routinely do now, and markers have helped us to fast track this issue. So where are we today with YAM improvement using at least genomics? We have whole genome sequencing and resequencing of D rotundata that is completed. Then we have whole genome sequencing and resequencing, where we also did chromosome or high C version 2 PacBio plus high C for D alata. And if you see, the total length is not a very large genome, it's a small genome. And we have other methods that we have used like any other crop, like whole genome resequencing, DART sequencing, genotype by sequencing, etc., for genotyping different populations. We have done, the GBS of 7 Guinea yam genotypes containing about 1,000 accessions. These are the different, this is the cultivated species, these two, which I have said that they are confused and they are still under domestication. So we tried to find out how there is, kind of how many, we got about 59,000 SNPs, GBS data, but it was not a very good, we had a lot of issues because we didn't know the ploidy of each of these accessions. So we faced some initial hiccups, but I think we had groups from EIB like Marcelo, Lindsay and others who were helping us to sort this issue. And we finally have some population structure and and the genetic diversity, and we could find that the cultivated and the wilds separated very widely, and we had a lot of duplicates. And if you see different populations, this is rotundata, and we can see that there's a lot of admixture in D rotundata, which is expected because it has mislabelings, it has D caiensis, and it has got other wild species also as admixtures. And this is how the PCA looked. But we also tried to see how we can differentiate with the PCA the different species, and for some of the species we had very few accessions, but in general we could find that TDC and TDR, that's what we call for rotundata and caiensis, they are together with Abyssinica. And there has been a lot of studies which says that Abyssinica is probably the progenitor for TDR and TDC, but separating TDR and TDC is still a nightmare for many of the for us when we try to identify the accessions or the breeding lines when we want to use them in the breeding program. So and then we have developed a SNP chip based on these studies, and we have now found a 46 to 50 SNPs which can work very well to define the population structure as well as for genetic diversity. We also have identified some of the traits for QTLs, like using QTL-SIC in D rotundata, and this is what is already published. And as I said, we do have, this is the female flower, this is the male flower, and that's how it should be. But sometimes we also find monoecious flowering in all the species mainly. Then we also use markers for sex determination, because this is so important in yam. At least we have seen in 2 species that we use very regularly in the breeding program, that is rotundata and alata. There is also switching of the species, the flowering or the sex. Like if this year you have seen them as female, the next year it switches to male. So, and then it switches back to female. So there is some kind of evolution going on that we are still yet to understand in flowering. Anthracnose disease, I think Agre has spoken about it very extensively yesterday, so I will not go for it. So we also do some, GWAS, and we have some qtls that we have identified for different traits, and these are also published. And I'll now come to some, what we did for high-C PEG biosequencing for D. alata, which is water yam. We also did some RNA sequencing, and we found 25,000 genes. And this seems to be one of the most complete whole genome sequence of Dioscorea species so far. We used about 10 crosses with 7 parents, about 1,500 genotypes or progenies to target qtls for different traits, and we used DART sequencing. And then we also tried to understand the relationship between these 7 genotypes or 7 parents, and which one contributed how much, and the IBDs, and we found some missing links when we tried to do the pedigree construction, and we tried to look at all the books and everything, the past books. We could not just find the contributor, the male contributor of this particular one. So there are some issues that we have in YAM, and I think simulation could be one of the best way to go forward. And we generated linkage maps, and these are the different map lengths. And then we also had a consensus map, genetic linkage map. Then for anthracnose disease, this was already shared by Agre yesterday. So this is how we score, phenotypic scoring, and we found some very good qtls. Interestingly, this qtl in chromosome 5 is the same that we found even with EST SSRs. So this is something we are really following and see if we can use it in marker-assisted selection. Then we do a lot of work with tuber traits, and you can see how much variation is there in tuber traits, and it's a nightmare to really do the harvesting. And but we use tuber flesh color using different chromometer and other visual ways, and then we try to do the normal distribution like any other crop. This is for the oxidation after 30 minutes, 60 minutes, and 180 minutes, and also for dry matter. And then we have found some qtls, putative qtls, which needs to be still validated for dry matter as well as oxidation, and different SNPs that control the qtls. Same with the different populations. This is one is showing with different population. So if you see, there are different populations that we have used from IITA, that's my institute, and also a national program called National Root Crops Research Institute. And then we have found different qtls and also their heritability. And for some of the traits like quality traits like dry matter oxidation, heritability is pretty low. And also some genes that we have identified, underlying these genes. And how do we do data management for YAM in general? So we have a project from Gates Foundation called Africa YAM, within which we have developed a database called YAMBase in collaboration with the Boyce Thompson Institute, and within which we have developed a software called YAMCross, where we use this very regularly for making our trials, our crosses. So everything is in the YAMBase, and then this YAMCross is developed by IITA in collaboration with BTI, and we can select the parents that we want to choose from, and then use them in the crossing. So in summary, I will say that there is a considerable progress that has been made in genomics-assisted breeding in yams. We have whole genome reference sequence now available for about 5 Dioscorea species. Some of them are in scaffold form, but for 2 species, that is rotundata and alata, which are the most important, we have now high-C reference genome sequence. Linkage maps, qtls, etc., both biparental and GWAS. QTLs have been identified for several traits. However, this is my issue that I have been actually asking people around, that we still face the issues of this mixed ploidy or in our populations, either mapping population, training population, or even parents, where we don't know what is the ploidy level. And this is our biggest challenge. And this may be related to the flowering also. So this is something we need to crack down. And I think I piggyback on this team of experts to help us to make sure that how we deal with something where we don't know the ploidy of the population or the progenies that we are working with. So with this, I'll go to the acknowledgement. We have the CGIAR program on root tubers and bananas, the National Science Foundation, and Africa Yam by Gates Foundation, which are the funding agencies. And honestly, there's a lot of support from EIB and Polyploid Group, and we are really thankful to all of you for helping us to crack a lot of issues in yams. And hopefully we will have some more funding and partnership in future to make sure that we can excel in this crop. Thank you very much.